Dov, check it's working.
Yeah, it seems to be recording. Yeah, brilliant. Thank you.
Okay, good afternoon everybody. My name is Kevin Shakespeare. I'm the Pro-Vice Chancellor for the Faculty of Science, and one of the great pleasures in my role is to introduce our inaugural speakers. An opportunity to celebrate a career as far as it's got so far, and also to welcome people who maybe don't often come to the university. It's lovely to see family and friends here today, and I think some people from Dov's career previously. So we hope you very much enjoy the presentation, and I'll be back to make a few comments at the end. So over to you, Dov.
Thank you very much, Kevin, and thank you everybody for being here. It's really humbling to see so many people here, colleagues, family, friends, people who've made some quite long journeys, so people who come from Johannesburg and Jerusalem, from various other cities, London, Birmingham, Cambridge, Oxford, even from University Park campus, even as far as our vet school, and even from both downstairs and upstairs in this building, so thank you all. I've also realised that I have left my phone on, with the ringer on. Now normally when I teach my first years, I do it deliberately and say, right, I've got to switch my phone off now, but I've done it accidentally now, so I'm actually going to put it on silent now. It's on.
It's here on the table. What's that?
Oh, that's my phone. So,
Absent-minded professor.
yeah, okay, just switch it on to silent. Right, excellent. So we're all here, or maybe I'm here, I don't know what you're here for, but the reason this event is happening is because I've been appointed as Professor in Computational Biology, which is kind of interesting given that my academic career did not start either in computation or in biology. My first degree was in mathematics. And there are various different branches of mathematics. There's some branches of mathematics that are to do with sort of solving problems in the real world. And then there's pure mathematics, which is the study of abstract things that are of interest to pure mathematicians. And that's how I started. And I was particularly interested in high-dimensional topological spaces and complex functions that can be defined upon them. And I have no intention of talking about any of these things, you're probably pleased to know, because they're not of great relevance. But actually, my interest in computational biology started long before then. It actually started as a teenager. And what I want to do is just talk through a little bit about my career, a little bit about the various important steps on the way, and give you a sense of sort of where I've come from, who I am, what research we're doing now, and I hope this will kind of work. So the first people to thank are actually my mum and dad, Julie and Ronnie, who are right here. And next to my mum and dad is my first love. So when I was 11 years old, they bought this computer. And every time I see an image of this computer, I'm still filled with this warm, fuzzy feeling. Okay? Now, I've no idea what other teenagers were doing. I didn't see them, right? I know what I was doing involved this computer. So I learned to program at the age of 11. And this has actually been really very important in everything that I've done since. So it's actually been really sort of central to everything that I've done. So that's the first point. And actually, my first forays into computational biology were at the age of 14. And the inspiration for this actually came from my great-grandmother, who is pictured here, and as you can see, I was always quite close to her. And I was a baby bear, yes. And by the age of 14, she was in her kind of final year of her life, and she was suffering from quite advanced Alzheimer's and was kind of forgetting who everyone was. And I was finding this quite challenging. And as the sort of person I was at the time, I did find it very challenging. The sort of person I was at the time, I decided to create a computer simulation of Alzheimer's. Now, not many people know this. No one in my family knew this at the time. but I actually programmed in something called a neural network into whichever computer I had at the time. This is a kind of a computing type algorithm that is used for machine learning. It's able to learn things. They're actually quite ubiquitous. They're all over social media, they're all over the web. People will use them for facial recognition, and I programmed in a fairly simple one, and it's a kind of a simulation of a brain. And normally you will use this for recognizing patterns. And so what I did was I trained it, because these kinds of computer programs can learn, I trained it to recognize a particular shape and a bit of an output, and give a particular output if that shape is shown. And then I kind of simulated the sort of the degradation of the neurons, the sort of gradual loss of function within this computer program as a simulation of what was happening inside my great-grandmother's brain. And gradually it lost its ability to recognise the shape that I trained it to do. And at the time I found this very comforting, and I say this was the very first piece of computational biology I did. Not related to that, at the age of 17, I became very interested in fractals. Now these are kind of mathematical structures that are, one part of it, a small part is similar to the whole. Now I've actually brought some real fractals along, and I actually need my helpers to bring some fractals around. Do I have 2 volunteers? I need some volunteers, okay? I need some volunteers, right? Yes, but I need you to hold this properly. Now, could you kind of pass this around to people to show them the broccoli so they can see how you can use that side of the room? And Rosie, could you have this broccoli? Okay, and you pass it around to people around there. So you go down this side and just let people have a look at it. And you go down this side, and what you can see. People the broccoli. Okay, and you can let them touch it. You can give it to them, and they'll look at it for a bit, and then they can do it. Right? Now, people who know me very well know I really dislike broccoli. Okay, I don't tend to eat broccoli. And but what you can see in this shape is that it's made of little parts that are actually look the same as the whole. And I wrote a computer simulation to produce this fractal third, and actually you may have seen it sort of appearing as I was speaking. And that's kind of I created that simulation recently, but it's exactly the same algorithm as I used when I was 17 years old. And for those who know, I'm not sure why, but at the age of 17 I was particularly attracted to the Markov chain algorithm that is actually kind of sampling from this kind of fern-like shaped space in a kind of in a particular way. I won't say any more, but at least several people will know what I mean by that. And so that was the second thing I did. Okay, so back to my undergraduate days. There I was an undergraduate at the University of Cambridge studying mathematics, and actually I'm really delighted that so many of my friends from that time are here, and it's a really, I'm really glad that you're able to make it. And I wasn't totally sure what to do with my maths degree, and that summer, by serendipity, I ended up having an opportunity to work in a cancer research laboratory, the Gray Laboratory, with this man, Boris Vojnovic. And so there I was, the very ideological 21-year-old me, in the lab photo, very excited that I was able to kind of use my mathematical skills to do something that was going to be of great benefit to humankind, and so off I went. And actually this is really very important, and I'm going to talk in some detail now about what I did that summer, because what you will see is that nothing really very much has changed since then. I'm more or less doing the same things, okay? So here we are. We were engaged, I say we, the laboratory was engaged, with types of experiments called pulse radiolysis experiments. And these are ways of trying to improve cancer radiotherapy. And in order to do these particular experiments, you need a high energy source of electrons, kind of a particle accelerator, and the particle accelerator they had was something called a Van de Graaff generator. Now, you may remember these from your childhood. They've got kind of like a big silver dome and they're kind of a belt, and when you put your hands on them, the electricity in them makes your hair stand on it. Okay, I was trying to get one. The physics department had one and said that their health and safety guys wouldn't let me have it. The engineering department had one, but then they kind of stopped communicating when I actually suggested that I might need to talk to another person, and then so that didn't happen. I even ordered online a kit for build your own Van de Graaff generator, but I ordered it too late and it still hasn't arrived. The broccoli arrived, that was good, but the Van de Graaff didn't. But this one was the size of a room, okay, and it would have these charged particles, and what you would have is a chemical sample. I'm out of frame. No, I'm not. Chemical sample. And what happens is the electrons collide with the chemicals, a chemical reaction happens, you have a type of chemical called a substrate, and it gets converted to a product, and as a consequence of this, and through these sorts of experiments, it is possible to... Thank you. It is possible to do things like do experiments on what sort of sizes of dose and length of dose and so forth, and understand the impact of this radiation upon the chemicals. And then you have a laser light that shines light of one colour into the chemicals, and then a light of a slightly different colour comes out into a detector, and then it goes into a computer, and that's where I came in. Boris was looking for a mathematician to help sort out the data. But what needed sorting out? So, here is a little simulation I've put together of what happens in these experiments. So each of these blue dots represents a chemical in its original form. They would really be whizzing around, but I didn't put that in the simulation. And during the experiment, there'll be a beam of electrons will kind of happen. That was the yellow flash. And then the chemicals start changing from their blue form to the red form. Okay, so they're kind of changing colour, and then gradually they'll all change. Now what we tend to assume, the kind of the science behind this, is that all these molecules, they're all the same. Okay, so although some change more quickly than others, this is just random, okay? They're all actually sort of changing at the same rate, but randomly some will move more quickly, some will change more slowly, and eventually they've all kind of changed into this red form. And then this is where the mathematics come in, because what was wanted to be known was basically how fast is this reaction? Now we sort of also need to know how many molecules there are, but that's just for technical reasons. What you really want to know is how quickly are they changing? Okay? And in order to do that, you construct a mathematical model. The model is simply a representation of what I've just said, this assumption that all these chemicals are changing from one form to another, and they're all roughly the same as each other. Some might go slower or faster, but they all kind of go at the same rate. And so you have the data, which are the black dots, and you have some kind of equation here, which represents what you think is going on. That's a nice simple equation here, and you can end up with this kind of red line that goes through the data. Well, that red line goes through the data only if you know how fast the reaction is and how many molecules there are. Okay? If you don't know those, you can't draw that line through the data, you don't know where to draw the line. So you've got to use computer algorithms to work out those two numbers, and in order to work out those two numbers, you can then get that line to go through the data rather than somewhere else, and then you'll know what you need to know. Okay? Now this process is a bit like climbing a mountain. Okay? And what was that?
You climb that.
I have climbed mountains. I haven't climbed this mountain. This mountain will be too difficult. It's not. It's the Matterhorn. It's the Matterhorn. So now basically there are a number of ways in which you can get to the top of the mountain. So and the computer algorithms work like this. You've got one way, which is you look at your feet, and you kind of look at where the steepest way to go is, and you take steps up in the steepest direction, and that's one way of doing it. You've got another way where you kind of look at the lie of the land, you take a guess of where the top is, and you take a huge, great big jump. The way I did it was some mixture of the two, which turns out to be quite a good way of doing it. There's an even better way of doing it, actually, which is to jump about at random, a bit like that fern, alright, in a special pattern, and you end up mapping the whole shape of the mountain, but never mind that. But that's what I did. And the consequence was, so there we go, the first scientific project I was involved with, 1992, Gray Laboratory Annual Report, very fuzzy, but you can see some lines going through some data, and that was the first thing that I was involved with, and although it wasn't sort of published in a peer-reviewed journal, it made the annual report. This software was used for about 20 years, and the facility was shut down. I say it was used for 20 years. Actually what happened was Julian, who's here, came 2 years later, looked at what I did and said, oh, you did it terribly, right? I did a much better job of it, and rewrote it. But never mind, it was the same idea, right? So So that was used for about 20 years. And as I said, this summer was really an inspiration for me because I came to realise that there were things I could do with my maths that were useful, that were relevant, that were interesting, that would do good things in the world, that there was a great need for people with these sorts of skills within the sort of biological world more generally, and I decided at this point that I was going to pretty much make my career in this area. So it was really pretty central. So I'm going to fast forward 20 years now, okay? There's 20 years of stuff is all going to happen in this slide, and I don't want to talk about it in great detail, but I will say that I did another summer job in a different cancer research laboratory. I went to do a PhD in Oxford, and I'm really delighted that where's Claire gone? That Claire is here from my PhD days. I went to work for Glaxo and actually ended up working with one of my old Cambridge friends, who's here, who's named. I went to work for Oxford Gene Technology, a biotechnology company. I wrote a book for Cambridge University Press, and all this was kind of going on, and that was all kind of fine. And then sort of some 15 years ago, I got my first academic job, which was at the University of Birmingham. And again, I'm really delighted that quite a few friends, both from the university and from the wider community in Birmingham, are all here. So John Heath, who was the head of school at the time, he appointed me to my first academic position. And when I was at Birmingham, I don't know how every person in their first academic job feels. It took me quite a few years to kind of work out what my group was about. What was my sort of who I was, what my research was about. And I had various PhD students, we did various different things together, and it took me sort of, a while to kind of get things going, so I'm not going to talk about any of that. Some of these projects were very nice, but I'm not going to talk about them. And then, I really want to thank the people who recruited me here, Joey Roberts, who was the head of school at the time, and Charlie Hodgman, who was here, who was the head of systems biology. That's Charlie CC. Who was the head of the Systems Biology Centre. And actually what happened was this advert came out once, and I was quite happy in Birmingham, and the advert came out once, and I just thought, it looks like an interesting job, but that's fine, I'm not going to, I don't want to come here, that's fine, I'm perfectly happy where I am. And then various people from Nottingham kept messaging me and saying, you really should apply for this job. And after the fourth time, when the advert came out the second time, I thought, well, maybe I should. Okay, and I'm very glad that I did. I had a wonderful time living in Birmingham, but actually, coming here from a research perspective has been absolutely the right thing to do. It's been very, and it's been a real pleasure to be here. So, what I want to do now is I want to talk about just two projects from my time here in Nottingham. The first of these is the very first project that was funded with me as principal investigator, as PI, research money I got in my own name, and the second one is the research that we're currently engaged with. Now I will say that I was PI on this particular project, but I can't claim credit for the idea or for the collaboration. So I'm really glad that John Hogman is able to be here. He's the person I kind of collaborate most closely, and my closest friend here. This came about through John's ongoing interest in metal resistance in bacteria. And I'll tell you a bit more about that in a couple of minutes. I became interested in metals and bacteria for all the wrong reasons, for some very boring technical reasons. But John has been interested in it because it's genuinely interesting. And John had a long-standing collaboration with someone called Taku Oshima in Japan. He was at the Nara Institute of Science and Technology at the time. And they came to me with a bit of a problem. You see, there was this funding call for working UK-Japan, and they said, we want to go for this call, but we've got two problems. First of all, it needs to be a new collaboration, and we've got an existing collaboration, we've had money together. Second of all, it needs to be what's called systems biology, which is basically, at the time, meant led by a kind of computational side of things. We need a systems biologist to lead it. So they said, could you kind of be in charge? I said, okay, fine. So I kind of edited what they wrote, we put it in, and we were very lucky. We got the grant. And actually this was brilliant. We got to work with these amazing Japanese people, in particular Hiroki and Taku, and Hiroki did all the modelling work here. Now why are metals and bacteria interesting? Why is zinc and E. coli cells interesting? Well, from a scientific perspective, zinc and many other metals are interesting because all of our cells need a certain amount of these metals in order to survive, but if they have too much, then they die. Okay? For this reason, cells, including every cell in our body, as well as bacterial cells, have evolved these mechanisms, molecular mechanisms, to import zinc when there's not enough, but to get rid of zinc if there is too much. Because too much zinc is toxic for cells, we use zinc as an antimicrobial. So it gets used, for example, in foot wash for cows and sheep. It gets used as antimicrobial feed for cows and pigs. It's even recommended for human babies as a way of preventing diarrhea. So you'll see some parts of the world where you'll see zinc tablets given to babies. And so you see, zinc is interesting. I won't talk about the details of that anyway. So we engaged in a project. Now the second reason, there are two things I want to say about this project. The first is it wasn't even my idea. And the second thing is, never mind all the equations and all the data, there was a very interesting story behind which I think for me tells a story about how science is done, how research is done. So Taku, in collaboration with John and Phil, who are both here, created these lovely E. coli strains which had bits of this system missing and which could glow in the dark according to what was going on, and generated this most beautiful, beautiful data. And Hiroki and I, we built our lovely, complicated mathematical model, which I said is a kind of a description of what we think is happening with all these genes switching on and off. And, you know, we couldn't quite get it to work. And Hiroki had kind of written this draft paper, but the model wasn't fitting the data. You see, we do the same things, right? I talked about the Grey Lab fitting the models to data. It's exactly the same process going on, okay? We're trying to get these dotted lines through these data, but look at this one, right? This is completely wrong, okay? Completely wrong, okay? It's in the wrong place, all right? We use the jumping around algorithm, by the way, for this one, all right? You jump around the mountain until you know what it looks like, okay? So there was a conference in Brussels, and we all decided to go. So John and his PhD student at the time, and Hiroki and me and some other people, we all decided we were going to go to the conference, and Hiroki and I agreed we were going to spend that week trying to sort this out. And actually this is one of the most enjoyable weeks of my scientific career. We didn't spend much time in the conference. Hiroki and I actually spent most of the time in the lobby of our hotel. Okay? Now I wanted to have a picture of us in the lobby of a hotel, but actually what was also going on was that Ada was exactly 1 year old, and Di and Ada came as well. And we were all staying in this hotel, and all the photographs we had was of us not working, but kind of playing with Ada and their videos of Ada using all the technology we're supposed to be using. And I thought I won't put up one of those. It didn't look like we were working very hard, but actually we were. Is that okay? And actually we were working quite hard, because what happened was this. Every morning, Hiroki and I would have a long discussion about what we think might be happening, and how we could model it, and what we could put in, and what we could take out, and we'd communicate with our microbiology colleagues, with John and Selena who were there, and with Taku over in Japan. And then in the afternoon, I went off with Di and Ada, and we just kind of, hung out in Brussels, which was very nice. If you haven't visited, it's a very good place. And then we went out for dinner, and then the next morning, Hiroki would turn up and he'd say, it doesn't work, model doesn't fit data. All right? And so we'd spend another morning doing the same thing. We'd have all these other ideas, and more discussions, and more conversation with the microbiologists, and we'd do this, and then off we went, we had fun, and we tried everything we could possibly think of. All right? We even tried not working for a day. We all went off to Bruges and had a nice time. Right? So, but that didn't work either. So we tried all of these things. None of it worked. Okay, and the last day I said, look, maybe we're just kind of thinking wrong. Okay, maybe this could never work because we've got the wrong sort of model. Right? Remember I told you about those chemicals? Okay, and I said that we assume they're all the same. What about if they're not all the same? Okay, now the way the experiment's structured, the way the diagrams are drawn, the way the model is created, all kind of assumes that all these things are all kind of doing the same thing. But what if they're not? What if they're all doing different things? What if some of them are doing one thing and some of them are doing something else? And so we took this hypothesis and it worked. So it worked as far as we could go. We tested it in various different ways with different types of models, with the cheapest and easiest experiment we could do, which also validated our result. There is a better and more complicated experiment, and one of John's PhD students is currently working on that. But basically it turned out, and we're pretty certain that we're right about this, that actually that kind of assumption that everything is the same is right. And I put on the previous slide those two terms, lost in the cloud and befriending the dragon. So lost in the cloud is a term used by the systems biologist Uri Alon for that state, where you've got no idea what's going on. You've got your data, you've got your models, and nothing works, and you don't understand it, and you get very frustrated and very kind of, it's a very difficult time. But I talk about befriending the dragon, which is the term to say, actually, this is the time of greatest creativity and discovery, because this is the point when you realise that how you're thinking about it isn't right. There's something fundamental about your thinking that's missing, that's wrong, and actually, if you're fortunate enough to kind of be able to step out of it, maybe the trip to Bruges did work, I don't know, and think about it differently, then you end up kind of having that breakthrough. It wasn't a big breakthrough, but it's kind of quite an important piece of work, and we think that this kind of reframes a lot of how we think about these sorts of this sort of biology. And so actually, this is actually not necessarily a bad thing, but actually quite a good thing. I shall now change tack to my final story. Oops, gone back. What's that too many? Right. So, there's another story of serendipity. So Steve Ransden, who I'm also glad is here. Where's Steve? There's Steve, right? One day, I say to Steve, can I visit the farm? Steve was the academic director of the farm at the time. We have a farm on our campus. It's a very lovely farm. As you can see, those who are visiting, we're in this beautiful rural location. We have a lovely farm. And Steve goes, sure. I just went out of curiosity. I just thought, I've been working here for a few years, I should go and see the farm. So we go to some fields. Right, there's some fields, there's some crop trials, they're very nice, there's some arable fields, very nice. We go to the dairy centre, which is kind of the bit of the farm that most people want to see. There's some happy cows, this is the dairy centre, the cows live in these sheds. There's some cows, they look like happy cows. There's what for most people is the highlight of the tour is the milking robot. There's this very high-tech milking robot where the cows milk themselves basically, and there's this kind of high-tech, I'm going to call laser-guided teat cups that kind of scan the cows, shake the cows' udders and kind of go on automatically and milk the cows, and it's all very nice. The cows are quite happy with this, they quite like it. And that's all very nice. And then I go around to the back of the farm where there is the slurry tank, and I get very excited. So I go back to my office and I call up John. I say, John, you've got to come and see the slurry tank. It's really cool. And then John and also Chris Dodds, who both with John and Chris are here, say, yeah, great, we'll come and we have another farm tour. And we all go around the farm, and John and Chris also get very excited. Now, our Vice Chancellor, Shira West, in her inaugural lecture, talked about what she regarded as a possible problem with universities, which she called normalised weirdness. Now, you see, the thing is that normal people, if they see 3 million litres of cow poo, okay, are likely to hold their nose and walk swiftly on. But I'm fortunate enough to work with perhaps weirder people, sorry John and Chris, who would see 3 million litres of cow poo and think, wow, that's brilliant, okay? Why is cow poo interesting? Okay, well, it's mainly fecal material, it's mainly water actually, but there's a lot of fecal material in there, a lot of bacteria in there. But there's not just bacteria in there. You see, dairy cows get mastitis. Actually, they get a lot less mastitis on our farm than they used to, which is a good thing. But dairy farms do get mastitis. And when they get mastitis, we have an obligation to treat them. I say we generally, the vets have an obligation to treat them, and they get treated with antibiotics. Dairy cows have other illnesses. So the calves may get respiratory disease, diarrheal disease, for which they get antibiotics. And there is also the antimicrobial foot wash. I talked about zinc. Okay, there's zinc and there's copper, and they go through and other chemicals. They go through the foot wash, and all these antibiotics. See, the milk from the cow with mastitis doesn't get sold to Tesco's with the rest of the milk. It goes in the slurry tank, right, along with its antibiotics. Okay, the foot wash goes into the slurry tank. Why might that be important? Well, at the moment we have a rather serious problem with drug-resistant infections. Okay, both within human medicine and within veterinary medicine we use a lot of antibiotics. About half the antibiotics used in the UK are in veterinary medicine or human medicine. And as a consequence of which, we have a problem with drug-resistant infection. I would expect that everybody here will know somebody who has had a drug-resistant infection. Okay? And of course, while a lot of the discourse that's rightly about what happens in hospital medicine, there is a part to be played with the fact that if half the antibiotics are being used in farms, then maybe that may be part of the problem as well. Now what happens in the slurry tank is as follows, okay? You've got all this mixture of bacteria, antibiotics, metals, all these things, and it's plausible to say that we could be selecting for drug-resistant bacteria in this environment. The slurry is very valuable. It's full of nitrates and other useful things, and it's used as organic fertilizer onto fields. So, all these bacteria and these resistance genes and these antibiotics and anything else that's in there goes into the soil, could go into our crops, get into watercourses, and is in the general environment. How important that is, we're not 100% sure actually. It's a matter of some quite considerable debate in the scientific community. But the question we wanted to know was, is there something to look at here? Now, I was very fortunate, or we were very fortunate. John and Chris had a new PhD student at the time, Delvine, and they said, Delvine, would you be happy to look at some E. coli from this tank? So she goes to the tank, she gets a sample, and she isolates some E. coli. The first E. coli that she looks at was resistant to 6 different antibiotics. Now this E. coli may or may not be harmful. Most E. colis are not. They live quite happily in a cow's rectum, they live quite happily in us, but some of them can be harmful, alright? We don't know if this is a harmful one or not, but what was important was there were 6 different antibiotics it was resistant to. Over her PhD she isolated about 150 E. coli strains, is that right? It's about 150 or so? And so she was working in other people's labs. That bit I did, I made the pretty picture, you see, and actually Dell did all the work. This picture, every row is a different strain of E. coli, every column is a different antibiotic. The big red block are all the drug resistances over there, alright? So there you can see that big red block, they're resistant to a bunch of antibiotics. This little bit over here is resistance to imipenem, which is a last resort human critical medicine that we found. Again, this appears and disappears on the farm, so it's a rather interesting situation. There's a whole long story in itself. What we got as far was, okay, there's a case to answer. Now, then at this point I was able to persuade the school to give me some money. This is quite good. The head of school gave me some money, right? The head of school gave me some money. So it wasn't me. It wasn't him, no. It was Neil. It was the previous head of school, right? And can I have some more money, son? And so we hired Michelle, who is here as well. That's Michelle, somewhere at the back, right? We hired Michelle, who produced a very simple model of what was going on in the slurry tank, and we have these models. We're not modelling chemicals or genes. Now we're looking at different types of bacteria. And what we wanted to understand was how these resistances can spread, and we got some interesting results, which again I'm not going to talk about. But the key thing here was, what we want to do is we want to try to understand what these risks are. Can we put numbers on them? Are there different ways that we could perhaps run the farm, or the way in which the plumbing on the farm works, literally the way the plumbing on the farm works, or which slurry you spread when, or which antibiotics you give to the cows, or how much of which antibiotics to the cow when, that might actually reduce the kind of resistance, or actually create more of a problem. And actually, this is the work that's ongoing at the moment, so I'm not going to say very much more about it, other than to say the following, right? We have even more complicated models with lots and lots of different equations in them. How many equations are we up to now? It's about 32 or something. We're still fitting models to data by jumping around the mountain in the manner of the fractal firm, and that's a very useful and a very useful technique. And this is ongoing work that we hope will be of value. Well, I forgot to say, I forgot one of the things I was going to say. I forgot the Lego slurry tank. The great thing about this work, though, is that we were very successful in getting some significant funding. And this funding was highly interdisciplinary, right? So what we ended up doing, is the reason why we were able to keep Michelle and various other people who are here, is because, now I need some helpers with this slurry tank, okay? You see, we have a Lego set here, and this represents the work that we do. Who wants to hold the Lego set? You need to walk it all the way around the room, this way and that way, okay?
Visualizer.
We could use the visualizer. I don't know if I can use the visualizer. charge of the Lego set, okay? And you see if you can see what's on the screen, okay? Now, so this poor chap has dropped his bucket. This actually happened and caused chaos on the farm, okay? You can see a vet. We work very closely with the vets. You can see social research. The farmer, looking rather definitely a farmer, right? Is being interviewed by somebody. You can see someone doing some computer modelling. That's Michelle. Okay. You can see a cow. Yes, you can see a cow. But we have the lab. We have the lab, right? We've got microbiology. So you can see some. It looks just like that, doesn't it? We have Lego Elf potion bottles, right? Yeah. Lego potion bottles at Alan Myers. And can we go to the mass spectrometer, please? Not only is this the only Lego slurry tank ever built, to my knowledge, you will also see the only representation of an Agilent LCMS mass spectrometer in Lego in the world, which is the black and white one right at the back. This one. This one. And also we have other projects. We have a PhD student with Helen who's interested in what happens when you spread the slurry on the crops and the soil. So we need this fields, all right. And then around the back we have another collaboration with vets who's interested in wildlife. There's a rat hanging around the back of the slurry tank, and he's been collecting poo from voles and mice and things. So it's highly interdisciplinary, and this has really been a fantastic way of working and a way of learning more about the world. And so that really kind of, okay, I'm going to go back to the slides now. So, press this button. Thank you, Adrian. Certainly when I was promoted, I suffered from a pretty hefty dose of imposter syndrome. Because there is a kind of a view of a professor as some sort of lone scholar who has got some great and deep knowledge about their subjects, and I don't really feel that way. But what I kind of hope to show you through these examples is the way in which my career has never actually been alone at all. It's always been in very close collaboration with others, working on projects that bring together different disciplines, and it's only by bringing together those different disciplines that we can actually make any kind of progress. And whether you're talking about cancer or drug-resistant infections, these are very sort of important, and it's the only way in which I have worked. So I don't actually know a great deal, really. I just kind of learned lots of languages and learned to talk to different kinds of people. So throughout my career, I've used different areas of mathematics, different areas of computation, but also worked with others within those fields who are more expert in those particular areas than myself. I've worked closely with biologists of various different kinds. The physical sciences have always come into it as well. And most recently what's been quite interesting is bringing in the social sciences, which has also been a very interesting and important revelation, and has really kind of improved many of the things that we do. So it's really very much a journey that is not a lone journey, but a shared journey and an interdisciplinary journey. And so for that, that's really good. So I kind of leave the final word, the final slide really, to thank all of my many collaborators. Now, not quite everybody on our collaborator with may be on here, and I apologise to anybody if their name should be on here and isn't, but there's obviously a wide range of different people, the people who mentors from my early career, people in my laboratory now. So Henry, I know I've talked about your work, but where's Henry gone? It is very interesting, and it is very important, and it's really good. People I've co-authored papers with, sometimes without any research funding at all. People I've done research with, got funding with. People from other people's labs that I've worked with, like Delvine and many others. And people in my own laboratory over the years with whom I have worked. There is one of my research collaborators is in red, that's Diane Levine. There are three things you need to know about Diane Levine. The first thing is, I talked about in my previous slide about working with social researchers. So Diane is the first social researcher I published with, and my only social research publication. So actually, that's how it all started, and it's actually Di who introduced me to the value of qualitative research, which for a mathematician may sound a bit strange, but actually maybe the other mathematicians realise that there's an awful lot of abuse of numbers, and sometimes you really don't want people to use numbers where they shouldn't. Qualitative research really steps in there as well. Second thing you should know about Diane Levine is she's brilliant at writing pathways to impact statements. So all those people who hate writing pathways to impact statements, you know.
Don't come and talk to me.
You can't talk to Diane unless you collaborate with the University of Leicester. But the most important thing you need to know about Diane is that she's my wife. and it's your love and support and everything through this journey would not have been possible without you.
We're
And I
super
love you
proud.
very much.
We love you.
And something else is going to happen. Two other people obviously bounce around my life. Ada and Rosie, who I couldn't possibly imagine my life without them. So thank you, everyone.
Thanks very much for a fantastic inaugural lecture. Congratulations and thank you to your two helpers who did a fantastic job. It's always fascinating to hear about people's career pathways and the different experiences that you've woven together, and as you quite rightly acknowledge, lots of people that have affected your career and helped you. And it's science, I've often said this in inaugurals, it's such a team activity and such a social activity, and you've really shown that really nicely today. It's also by far the best use of Lego and the visualizer. I think we were all sat there as you were trying to turn it on going, it's not going to go on, it's not going to go on. And it was like this shock as it actually came on. And then the beautiful guided tour of all the different parts on it was great. I suspect not Lego's biggest selling kit.
You can propose it. If enough people vote for it, Lego will have to make it.
So just tell us a little bit about that. Who actually put that all together? It must have been...
At some point, the prototype was repurposed as a swimming pool for mermaids, and then the inside part of the slurry was referred to as a chocolate cake for a while.
And you got a real chocolate cake.
And there is a second representation of the slurry tank that will be downstairs shortly. Thank you.
So, again, thank you very much. Fantastic talk. Congratulations on everything you've achieved. I know you're going to achieve much more in the future. I think we're going to go downstairs and have a drink. Is that the idea?
So yes, there should be tea and coffee and cake and other things downstairs.
Fantastic. So we'll just ask you all just to congratulate Dov again and join us downstairs. Thank you.